Welcome. In this session, I'll continue discussing about nucleases. Then after which I will also discuss about how the first recombinant DNA was constructed, and we'll also see about what actually is this gene cloning and gene transfer. So in the last session, I have told you that the nucleases, so these nucleases are enzymes which are present inside the nucleus, and these nucleases are classified into two types. One I said exonucleases and another one called endonucleases. And I told you that exonucleases will remove nucleotides from the ends of DNA molecule. So they are not going to remove the nucleotides, okay, at the center or somewhere else within the DNA molecule. They are going to remove, okay, from the ends of the DNA molecule. Say suppose here I have taken a DNA molecule, okay, so 3 dash and 5 dash, and you can see here this is a nucleotide and this is another nucleotide and so on. So I hope you remember the structure of the DNA molecule and say these are the nucleotides. Now the exonucleases are going to remove these nucleotides one by one. So say first this it has removed this, then later it has removed, and it will be going on, okay, in a direction. So that will be from end of the DNA molecule, not within the DNA molecule. So coming to this endonucleases, so in this case they are going to cut DNA, or they are going to cut at a specific location within the DNA molecule. So I hope you understood the difference between the exo and endonucleases. So here they are going to cut at a specific location within the DNA molecule. Say here I'm going to take a DNA molecule, and when we use this restriction endonuclease, so it's going to cut at this point and at this point. And I already told you that these nucleases will be breaking only the phosphodiester bonds, not N-glycosidic or the hydrogen bonds that exist within the DNA molecule. They will not alter any of those bonds except the phosphodiester bonds, right, thereby separating the DNA molecule. One example of these endonucleases is that restriction endonuclease. So it's a restriction endonuclease. In short, it's called REN. And I told you in the last session that the first REN that was discovered is HIND2. Okay, and after that we have discovered more than 900 restriction endonucleases from, okay, 230 strains of bacteria. So from these varieties of bacteria we have isolated more than 900 restriction endonucleases, and each restriction endonuclease that we have isolated, we have given it a specific name. So that's the nomenclature of RENs. So how we will be giving names to those restriction endonucleases that we isolate from the bacteria. Say suppose here I'm going to take echo R1. So this is the name of restriction endonuclease, and this restriction endonuclease is obtained from a bacterium called Escherichia coli, right? So this is the scientific name of that bacterium from which we obtain this restriction endonuclease. Okay, so this is the name of restriction endonuclease. And if we see in that name restriction endonuclease, you can see the first letter of the genus. Okay, so this is the first letter of the genus. So in the restriction endonuclease, okay, name, the first letter corresponds to the first letter of the genus. Then coming to the next two letters, the next two letters will be the first two letters of species, right? And then coming to the R, so this R stands for the strain. Okay, and coming to that Roman number 1, so this Roman number, so we will be giving Roman numbers to these restriction endonucleases in the order of their discovery from that, okay, bacterium. So in this case I have mentioned here Roman number 1, which means that this is the first restriction endonuclease that I have obtained from this bacterium called Escherichia coli. Okay, so assign Roman numbers in order of their discovery. So will be assigning the Roman numbers in the order of their discovery here. Okay, now I have one question. So if I give the order in which we discover or isolate the restriction endonucleases from that bacteria, then why the first restriction endonuclease name is not Hind1? So why I have given it Hind2? Okay, you find it out. Okay, so I hope this is clear about the restriction endonucleases and how we will be giving names to those isolated, means the restriction endonucleases which we will be isolating from those bacteria. And I told that these restriction endonucleases will cut the DNA at specific locations. So how the restriction endonucleases cut the DNA at specific locations? So that we'll see that, okay, RNAs, okay, identify something called palindromic sequences. So they are going to identify palindromic sequences, okay, and they are going to cut within that palindromic sequences. So now question comes, what are these palindromic sequences? So palindromic sequences are the bases that will be read, okay, same, okay, from the left to right and right to left. So that I can say from 5' to 3', okay, that is from the left to right, and again from right to left. So say I'm going to take from 5', say suppose GAATTC. Of course, the same nitrogen bases here, guanine, adenine, thymine and cytosine. And when you read from this to this side, from left to right, it is GAATTC. And when you come from this right side to again to the left side, again it will be GAATTC. So those sequences which will be read same, okay, from both the ends are called palindromic sequences. So these restriction endonucleases are going to identify such palindromic sequences and going to cut, okay, the DNA. Now if I am taking this echo R1 as an example, so this echo R1 is actually obtained, as I told, from Escherichia coli RY13. Okay, that's the complete name of this E. coli. So E. coli RY13. So that's what we have mentioned in that way, the name echo R1. And this echo R1 is going to cut between the G and A. it's going to cut between G and A. And you have to keep in mind here that each run identifies a specific palindromic sequence. So each run identifies a specific palindromic sequence. It's not like the echo R1, okay, can identify another palindromic sequence, not at all. So this echo R1, if I use, then it will identify only this sequence. If that sequence is not present within the DNA molecule, it's not going to do anything. If it is present, then definitely it will cut between the G and A, thereby separating, okay, the DNA molecule. Okay, I hope that's clear. So I have to keep in mind that each restriction endonucleases identifies a specific palindromic sequence. So it means that every restriction endonuclease has its own, okay, particular palindromic sequence. It will identify only that palindromic sequence. If it is present, then it will recognize and then will cut accordingly. Okay, and don't think that every time a restriction endonuclease will cut between G and A only. If the some combinations may cut between T and T, between T and C, between A and T. So there is several combinations. Okay, so we have more than 900 restriction endonucleases with various combinations with various palindromic sequences, and cut points will be also different. Now let's see, okay, how these restriction endonucleases, I have given that they will be identifying particular palindromic sequences. Okay, so when we use this restriction endonuclease, so it is going to cut the DNA at a specific location by identifying a particular palindromic sequence. Okay, now when the restriction endonuclease cuts the DNA, it will produce, okay, two types of ends. So there will be two types of ends in a DNA molecule after using this restriction endonuclease. So what is that? So some restriction endonucleases are going to produce something called blunt ends, also called the flush ends. So they are going to produce blunt ends or flush ends. Then in another case, they are going to produce sticky ends, also called cohesive ends. Right? So when I use restriction endonuclease, I told you that there are several types of restriction endonuclease. Each restriction endonuclease will identify a particular palindromic sequence. It may cut within the palindromic sequence, okay, on both the strands at the same point or maybe at different points. So first, let's see, in this case, the blunt ends. So how they are going to produce blunt ends? Say suppose an restriction endonuclease named, okay, ALU1. So now this restriction endonuclease, which is ALU1, is going to identify a particular palindromic sequence that will be AGCT, and say this is the polarity, okay, and from this end it will be again AGCT. Okay, for the complementary also we need to see. Okay, this is the ALU1 restriction endonuclease. So once the ALU1 recognizes this palindromic sequence, it is going to cut between G and C again on one strand. So we know that this is one strand of the DNA and this is another strand of the DNA. So we know that DNA is a double stranded, right? And what this ALU1 does is it's going to cut on one strand between G and C and also on other strand between G and C. As a result, what you will get is the blunt ends. So this is AG, then CD. Right. So these are the blunt ends. Okay. So that's one type when we use restriction endonucleases. Now coming to the second one, sticky ends or cohesive ends. In this case, I'll take the same example, echo R1. So this echo R1, already I told you, it will identify the palindromic sequence GAATTC. And okay, from this direction also same, the complementary G. For this T, it is AATTC. Right, but here it's not going to cut at the same points on both strands. So in this case, the ALU1 cuts at the same point on both strands. So it cuts at same point on both strands. So because of that, you will get blunt ends. So in this case, it won't be. So what happens here, it will cut between G and A. So it's going to cut between G and A. So echo R1 recognizes this palindromic sequence first. Then it cuts between G and A. So between G and A, so what it cuts is that, okay, or it breaks phosphodiester bond. So there, what it becomes is G A A T T C. And in this case, it will be A A G. So you can see here, this echo R1 does not cut at the same points. So it cuts the DNA strand at different points. You can see here, but it will be cutting between the same bases, that will be guanine and adenine. Right, because of that, you can see a protruding end. So you will get a protruding end. So these protruding ends, what we call them, the sticky ends. So why they are called sticky ends? Because they will be having, okay, the hydrogen bonds which can readily form pairing. So for that reason, they are called the sticky ends or the cohesive ends. So these are, okay, two types, okay, of ends. So two types of ends in a DNA molecule after using the restriction endonucleases. I'll say two types of ends in a DNA molecule. So in DNA molecules after using restriction endonucleases. Right. I hope we understood that how the blunt ends are formed and how sticky ends are formed. Don't think that this echo R1 will never form the blunt end, nor that ALU1 forms this sticky ends. So I told you that this echo R1, it is fixed. It will recognize only this palindromic sequence. Like that, whatever that 900 restriction endonuclease, each one of will be having its own palindromic sequence to be, okay, identified, and will have its own point of cutting. So some may form blunt ends, some may form sticky ends. So it's our choice while making recombinant DNA. So what kind of restriction endonuclease you want, so whether you want the sticky ends and the blunt ends. Mostly we choose with these sticky ends, we'll go with these sticky ends. Right, as these restriction endonucleases are able to cut the DNA, so we can also call the restriction endonucleases as molecular scissors. Okay, so RENs are molecular scissors. So now how I'm going to use this restriction endonucleases and how I'm going to isolate a particular segment of DNA from an organism, and that how I'm going to introduce into another organism. So let's see here. I'll just give an example. So say this is the DNA molecule of an organism which I have isolated, and what I'm going to do, I'm going to cut this DNA molecule, and I need to isolate a specific segment of this DNA molecule, and for that I'm going to use something called some restriction endonuclease, okay, some restriction endonuclease I have used, okay. And then the organism, so this is another organism, so this is another organism's DNA. So I need to, cut a specific portion of this, DNA segment, and I need to insert into this DNA. So this is DNA of host organism, so DNA of host organism, okay. And this is the uh, DNA of source organism, right? And now to put this DNA into this, okay, DNA of the host organism, someone has to carry this, okay, to this point, and that is what I call a vector. So a vector is required here, okay. Specifically that is called a cloning vector. It's called cloning vector, and an example of that cloning vector is plasmid, okay. So these plasmids, some of the bacteria naturally have these plasmids, right? So they are double stranded DNA molecules, and they will be circular and are capable of self-replicating. So a plasmid is a double-stranded DNA molecular which is circular and has ability to self-replicate. Okay, so this is how we will be seeing, okay, the DNA molecule. Of course, here I don't think it is linear, so it is still, it will have that, you know, the double helix model. So you will be seeing still something like this. So this is the plasmid. So it is double stranded, okay, and it is circular. So the ends are joined. Actually, you may find two types of DNA molecules in the organisms: the linear forms or the circular forms. The linear forms, here you can see these two which I have drawn are linear. The ends are not joined. But in this case of the plasmids, so we see that ends are joined together to form circular. And these plasmids are found in some bacteria. So they are found in some bacteria. Not all bacteria will be having plasmids. Some bacteria will be having these plasmids. Okay, and this is what I call the cloning vector, and that's what I have mentioned. So here first I need to put this the selected, or what I say this as my interest, so hence I call gene of interest. So this gene of interest I need to, okay, link with this vector. Okay, I need to link with this vector, okay, and then use this vector to deliver this gene of interest into the DNA of the host organism. So that's what here I'm going to do. So I'll make a cut, and then I'm going to introduce that right? So this is what I say, in fact, the rDNA. So I can call this as rDNA, the recombinant DNA molecule. Okay, and that I'm going to put it into the DNA. So this vector is going to carry this gene of interest into the DNA of the host organism here. Now to do this, for all these, for the vector, for the source DNA of the source organism and DNA of the host organism, for all that I need to use, okay, same restriction endonuclease. So need to use same restriction endonuclease. So only when we use the same restriction endonuclease, all of them will have the same ends and it becomes easy for them to join. So if you are going to use different restriction endonuclease, we know that different restriction endonucleases will identify different palindromic sequences and they will have different ends, and those ends will not be complementary to each other. So for that reason, they will not join. Okay, of course, don't think that they will be joining by themselves. We need to use again other enzymes like DNA ligases. So the DNA ligases are the enzymes that we need to use, okay, to join this gene of interest into the vector or into the host organism's DNA, right? So that's how we use these restriction endonucleases to cut the DNA, okay, of the source organism and select that gene of interest and then link it with a vector and use this vector to deliver that gene piece of gene into the DNA of the host organism. Now let's see, okay, how the first artificial recombinant DNA molecule they generated. Okay, so here it happened in 1972. So it happened in 1972 and that was done by two persons, Stanley Cohen and Herbert Boyer. So Stanley Cohen and Herbert Boyer are the two persons who actually constructed an artificial rDNA molecule. So they constructed, okay, artificial rDNA molecule. Okay, so constructed artificial, they constructed first an artificial rDNA molecule. So how it became possible? Of course, that is all became possible by using these plasmids and then restriction endonucleases and then DNA ligases. So here I just mentioned that this DNA ligase, okay, will help in formation of, okay, phosphodiester bonds. They help in formation of phosphodiester bonds, thereby helping in joining two fragments of DNA. Say here I have a fragment of DNA, right, and it has a sticky end. So this portion is the sticky end. Now I'm going to take another one that has also a sticky end. Has the sticky end. So here this portion is the sticky end. Now if I have cut these two DNA molecules with the same restriction endonuclease, then they will have base complementarity. Okay, so say suppose here it is CGAT, then here it will be ATCG. So you can see there is a complementary between these two sticky ends, then they can join. So you can see the phosphodiester bond formation, and that can be done with the help of this enzyme called DNA ligase. Okay, so that's what with all those tools, this Stanley Cohen and Herbert Boyer, first they have constructed an artificial recombinant DNA molecule here. Okay, so what they have done is they have isolated a gene that is coding for antibiotic resistance. So a gene coding for antibiotic resistance. Usually such genes which actually help in giving antibiotic resistance are present in plasmids. So they are present in the plasmids and give the bacteria antibiotic resistance. So say suppose if I say there is an antibiotic called tetracycline, and this plasmid has, okay, tetracycline resistant gene. Now when I put that bacteria having the plasmid, and that plasmid contains, okay, tetracycline resistant genes. So if I put that bacteria in tetracycline, the bacteria will not die, because the bacteria has plasmid, and that plasmid has tetracycline resistant gene, and that will resist the effect of tetracycline. So that is what the meaning for antibiotic resistance. So here I hope if you recollect from the first year when we have studied, okay, the bacteria will be having these plasmids, and those are extra chromosomal DNA molecules, and they're not part of the bacterial genome, right? So this is the bacterial genome, and these are all plasmids. So a bacteria may have only one plasmid, or more than one plasmid it may have. Okay, and if it is the case, then all those plasmids will be of same type, means they are all copies. So because I told you the plasmids are self-replicating, right? So I'll tell that point. So here the Stanley Cohen and Herbert Boyer, so they have isolated a gene that is coding for antibiotic resistance, and what they have done is they have linked this gene, so linked this gene to the plasmid of so they and linked this gene with native plasmid. So the plasmid belongs to a bacterium called Salmonella typhimurium. Is the name of the bacterium, Salmonella typhimurium. So they have linked this antibiotic resistant gene with the plasmid of Salmonella typhimurium. Okay, say suppose I'm going to show you that. So this is the I have taken this, and by using restriction endonuclease, I have cut this the gene of interest, and say this is antibiotic resistant gene. It's antibiotic resistant gene, and now the plasmid of Salmonella typhimurium. Okay, so this is the plasmid of typhimurium. So what I have done, so this one by using the same type of restriction endonuclease, I told you we cannot use two different restriction endonuclease. Then if it is the case, it will produce two different sticky ends which will not be complementary, right? So this I'm going to use the same, and then I'm going to insert this antibiotic resistant gene into here. Then this is what I say, the rDNA molecule, right? So is that clear? Now, okay, to express this whatever that antibiotic gene, so it requires, environment, and that is inside the cell. So here we know that this antibiotic resistant gene to get expressed, right, it means to produce a product that can counteract the antibiotic action, so it should get expressed. So that expression can be done only after you put this plasmid into a host organism, and that host, here we will take bacteria. So here I have kept this plasmid into a E. coli bacterium. So what I have done in this case, so this plasmid I have introduced into a bacterium. So this is the genome of that bacterium, and you can see here it has that antibiotic resistant gene. Okay, so now it will start expressing inside, and at the same time, when this bacterium multiplies, okay, then you can see it produces, okay, when it multiplies, and along with that, this plasmid, so this is the rDNA actually, right? This rDNA will also get multiplied. So that will also get multiplied. I'm just mentioning, okay, in this way. Right, so you can see, because this all will be remaining same, so this rDNA will also get multiplied. So this is what we call it the gene cloning. It's called the gene cloning. Okay, so after making this, rDNA, I have introduced into a bacterium and allowed that bacterium to multiply, and when it is allowed to multiply, the rDNA present inside will also get multiplied along with the bacterium, and whatever the number of copies of the bacteria will form. Okay, so we know that they will multiply uh, in a geometrical fashion, so 1 2 4 8 16 32. So after few generations, so just 1 2, okay, 3 4 5 generations, you will have, okay, 32 copies of this rDNA, and that process what we call it gene cloning, right? I hope that part is clear. And next, I can even use these vectors. I can use these vectors even to deliver that gene of interest into that host organisms, where I can say it as the gene transfer, that we will see in detail in the coming sessions. Right, now here, after linking this gene of interest with the plasmid, okay, so I told you that the plasmids are self-replicating. So how they are self-replicating? Because they will be having something called origin of replication. So here we should ensure okay, that the plasmids have that origin of replication while constructing. Of course, we know that we are able to construct these plasmids. Once we take a plasmid from another bacterium, remove unnecessary genes, whatever the genes we don't want, we can remove them and then replace with some other genes, or you need not replace that with another genes, whatever. So you can reconstruct. So after taking that plasmid, eliminate the genes what you don't want, and make it in such a way that it is capable of delivering the gene of interest that what you are going to link it with, so with that plasmid. Okay, so later I will discuss especially a plasmid named PBR322, how they have constructed and what are all those restriction sites it has on it. And so I have not yet finished discussing the cloning vectors in detail. I'll discuss that cloning vectors in detail in coming sessions. Okay, so these plasmids, okay, or to any if you want to make the copies of that gene of interest, you must put into a DNA that should have origin of replication. So in short, that is called the ORI site. It's called ORI site, so which means the origin of replication. So without this, right, these plasmids or any other DNA molecule, even though if you have linked foreign DNA with it, if it doesn't have that ORI site, it will not going to multiply. So it must have this origin of replication. We know this origin of replication will have some specific base sequences that will be identified by polymerase enzymes, and those polymerase enzymes, after identifying it, so they will help in, okay, replicating the DNA molecule and making the copies of the DNA molecule. Right, so with this, I will end today's session. In the next session, I'll be discussing the diagrammatic representation of our DNA technology, and we also see that the separation and isolation of those DNA fragments. So how I'm going to get that gene of interest and all there is a procedure for that. We'll see that in the next session, and also we'll see about the cloning vectors in the next session.